Imagine your brain is like a car. During adolescence, the gas pedal works great. You're ready to go, try new things, take risks. But the brakes, they're still developing. And if your brakes aren't fully working, things can get dangerous pretty quickly. That's kind of what's happening in the adolescent brain. Today, we're going to explore what's going on with the brake system, especially when it comes to nicotine and alcohol, and why it doesn't fully develop until adulthood. In our previous episodes, we've talked about how adolescence is a time of increased risk-taking and novelty-seeking. It's also when the brain is undergoing a lot of rewiring, especially in systems related to stress and reward. Because of that, adolescents tend to be more sensitive to drugs like nicotine and alcohol. What's really interesting is that nicotine and alcohol actually have a bidirectional relationship, meaning using one can enhance the effects of the other. Because of this, researchers often studied them together. In fact, previous studies have shown that adolescent male rats are more likely to self-administer drugs when given both nicotine and alcohol compared to either one alone. So clearly, something about the adolescent brain is making it more vulnerable. That brings us to today's focus, a specific brain pathway that might help explain why. Welcome to the Translational Addiction Podcast, where we explore innovative research from the Lotfipour Lab at UC Irvine, dedicated to advancing translational addiction science and developing more effective treatment strategies. I'm your host, Ishva Patel. I'm a business administration and biology major at UCI, and I'd also like to welcome our co-host.
Hi, I'm Arshiya, and I'm a first-year biomedical engineering major at UC Irvine. Today we'll be discussing the paper Dynorphinergic Lateral Hypothalamus to Posterior Ventral Tegmental Area Pathway Matures After Adolescent in Male Rats by Alexandra Rogers, Emily M. Castro, Shardad Lotfipour, and Francis M. Leslie. The study looks at the connection between two parts of the brain, the lateral hypothalamus and the posterior ventral tegmental area. The VTA, or the ventral tegmental area, is located in the midbrain and is defined by a cluster of neurons. This area of the brain plays a critical role in the brain's reward system, motivation, and addiction. The lateral hypothalamus, on the other hand, is located in the lateral zone of the hypothalamus and projects neurons into the ventral tegmental area, also contributing to reward-related behavior. The VTA is still developing during adolescence, so researchers wanted to see if the pathway between the lateral hypothalamus and the PVTA might explain why adolescents respond differently to drugs. Looking a little more deeper into the brain, there are receptors called kappa opioid receptors, which bind to a naturally occurring molecule called dynorphin. When dynorphin activates these receptors, it usually raises the reward threshold, essentially making things feel less rewarding to the individual. It is also important to note that when dynorphin is released, orexin is also co-released by many neurons. Interestingly, they have opposite effects on reward threshold. For example, when kappa opioid receptors bind to dynorphin, they become active and increase the reward threshold. But studies have also shown that orexin in the VTA also drives behaviors motivated by drugs. Orexin expression differs by age and may have behavioral impacts, which is why this study also focuses on orexin expression in different study groups.
Previous research adds another layer to this. One study found that blocking kappa opioid receptors made adult male rats less sensitive to the reinforcing effects of low doses of nicotine and alcohol. Another study showed increased activity in the lateral hypothalamus to the posterior ventral tegmental pathway when adult rats were exposed to these substances. So when you put all of this together, it suggests that the kappa opioid receptor system in the posterior ventral tegmental area might not be fully functioning in adolescents. And that could be a part of why their brake system isn't as effective, making them more susceptible to nicotine and alcohol. With that, let's take a look at how the study was conducted.
In this study, the researchers used only male Sprague Dawley rats, as earlier works have shown that the combined effects of nicotine and ethanol were much more prominent in males. Some rats arrived as adolescents, while others arrived as adults, so researchers can compare how age influenced the brain's response to this specific drug combination. The adolescent rats stayed with their mothers until weaning, while older rats were housed in a group with access to food and water. The rats were handled every day by the researchers before the experiment to get them accustomed to human contact in order to remove the variable of stress in the experiment. The brains of the rats were then preserved and sliced into thin sections. Researchers then stained them with several different antibodies. One of these antibodies helped identify prodynorphin-producing neurons so that researchers could mark dynorphin-related cells, which are involved in stress, reward, and drug-related signaling. Another antibody labeled the protein CFOS, which acts as a marker of recent neuronal activity and shows which cells became active after the nicotine and ethanol exposure. Lastly, they used fluorogold to label and identify neurons that were connected to the PVTA. After staining, the researchers counted the total number of prodynorphin, or PDYN, positive neurons, the total number of CFOS positive neurons, the number of neurons positive for both PDYN and CFOS, and the number of fluorogold labeled cells. These cell counts were done manually using the software ImageJ. The researchers were blinded to which treatment group each rat belonged to in order to reduce experimental bias. Finally, the data was analyzed using a two-way ANOVA to compare the effects of both age and drug treatment. Post hoc tests were then used afterward to determine which specific groups differed significantly from one another.
Earlier, we talked about how the researchers believe that inhibition of the kappa opioid receptor and potentially a blockade of dynorphin signaling might be what is leading to increased susceptibility to reinforcement as observed in the adolescent mice. Hence, when analyzing results, the researchers looked at what was happening in the brain after nicotine and alcohol exposure. Specifically, they measured the number of dynorphin-positive neurons in the lateral hypothalamus and the dynorphin and CFOS positive neurons to see how many of those neurons were active an hour after nicotine and ethanol administration in adult and adolescent rats. The researchers found that the number of dynorphin-positive neurons were consistent between the saline and nicotine and alcohol-treated rats. But when comparing age groups, the number of dynorphin neurons per sample was higher in adults than rats. It was also observed that the dynorphin-positive neurons, which were also CFOS positive or active, almost doubled in adult rats that received nicotine and ethanol. Both of these findings show how age plays a major role in responses to drugs. Furthermore, researchers also looked at how the pathway between the lateral hypothalamus to the posterior ventral tegmental area develops. They injected fluorogold into the posterior ventral tegmental area and quantified the number of fluorogold-positive neurons in the lateral hypothalamus to visualize the connecting neurons. They also calculated the prodynorphin-positive and orexin-positive cell counts. They found that adults had more cells labeled with fluorogold and prodynorphin, while adolescents had significantly more cells labeled with orexin, suggesting that the dynorphinergic circuit between the lateral hypothalamus and the posterior ventral tegmental area develops later in life and is more active in the adult brain than the adolescent brain.
A major takeaway from this study is that adolescents don't have the same biological pathways as adults. Important reward-related circuits are still actively developing during this period, and those differences may help explain why adolescents are more vulnerable to nicotine and alcohol use. In adults, dynorphin signaling may act like a brake that reduces how rewarding nicotine and alcohol feel. Since adolescents don't seem to have this fully developed pathway, they may experience drugs as more reinforcing. This study also highlights how complex the balance between dynorphin and orexin may be. Dynorphin is generally linked to aversion and reduced reward gratification, while orexin is more associated with positive reward and motivation-seeking behaviors. Since adolescents had fewer dynorphin-related projections but more orexin-positive cells, the balance in the adolescent brain may be shifted towards more reward-seeking behaviors. That imbalance could make young individuals more susceptible to addiction-like behaviors. Although this study answers important questions, it also opens the door to many future directions. Future studies could examine multiple ages between adolescent and adulthood to figure out exactly when this dynorphin pathway develops. Researchers could also investigate whether the same pattern exists in female rats, since this study only included males. Another important next step would be testing whether blocking kappa opioid receptors, specifically in the posterior VTA, changes nicotine and alcohol reinforcement in adults. This would help determine whether the LH to PVTA dynorphin pathway directly causes the behavioral differences seen between adolescents and adults. Finally, future studies could look at other brain regions, such as the amygdala or nucleus accumbens, to better understand how the broader reward network develops over time.
Ultimately, this study brings us one step closer to understanding why adolescents may be especially vulnerable to substance use. By identifying a specific reward-related pathway that develops later in life, the researchers provide a clear explanation for why nicotine and alcohol can feel more reinforcing during adolescence. Understanding how these pathways mature could eventually help researchers design more targeted prevention strategies and treatments for addiction. If you're interested in learning more about the effects of nicotine across a lifespan, follow us on Instagram at Translational Addiction Podcast for updates, and check out the infographics and papers on the Lotfipour Lab website linked in the show notes. If you or someone you know is struggling with addiction, you are not alone. Please contact your healthcare provider or refer to the resources linked in the show notes. The Translational Addiction Podcast is a production of the Lotfipour Lab at UC Irvine, exploring how to understand addiction and develop effective treatments. See you next time, and thank you for listening.